Okay, proteins. This macromolecule, or organic molecule if you prefer, we have quite a few functions here. You have quite a lot of different types of proteins in your body. Some of them are enzymes. We've talked about enzymes being involved in chemical reactions. They allow us to do chemical reactions faster by lowering activation energy. We said we can do reactions without enzymes, but we just prefer to use enzymes because they get to go faster and we get to use less energy, and that's always nice. Many of your proteins play a structural role, so your cytoskeleton inside of your cell, okay, some of the components that help give your cell a specific shape. Some of these are proteins. Your muscles, okay, are full of protein molecules. Okay, so it helps us give us the overall shape that we have. Some proteins are involved in movement. Okay, so those muscles I just mentioned, not only do they give us shape, but they also allow us to move our bodies. So your actins and your myosin proteins pull on each other, and that's how we get skeletal muscle movement. Some proteins are involved in your body's defenses. So if you've ever heard the term antibody, okay, antibodies are technically a type of protein. Now, we can use proteins as a fuel source, okay? And we actually can use fats or lipids as a fuel source as well. But notice we haven't really made a big deal out of that. It's because your body has a little bit harder time breaking down proteins and fats for energy. There's a couple extra steps involved to get the energy out of these types of molecules. So obviously, unless you're on a special diet, you are probably not just gorging on carbohydrates, as delicious as they are. You are probably having your meals contain proteins and lipids as well. So what does your body do with those? In addition to, the enzymes, the body defenses, yes, we are using them for fuel as well. Your body just loves carbs extra hard because those carbs are easier to break down for fuel. Okay. Now your proteins, these monomers are amino acids. Okay, and that's our little monomer. You technically have 20 different types of amino acids. They range from very simple glycerin, okay, to really complex things like tryptophan. You will be happy to know that you do not need to memorize all 20 of these by name or by sight. We just want you to know, hey, there's 20 different types of these, okay? And we also would like you to know that each amino acid would be attached to the next amino acid. Boop, boop, boop. Okay, so if we linked these, if we chemically bonded them together, we would be forming peptide bonds, okay? And it is a type of covalent bond. And when we start linking up these amino acids through peptide bonds, you are creating a polypeptide. Okay? Polypeptide chains are then modified into fully functional proteins. Okay? Now, we just mentioned attaching these through peptide bonds. How are we going to physically do that? We are back to dehydration synthesis reactions. So we have our alanine amino acid, we have our glycine amino acid. We would like to bond these guys together. So we are going to take a little OH, a little H, remove all those. Okay, H's and OH's make water, so we have dehydrated these molecules. We have removed water, and when we do that, we made room to form a peptide bond. Okay. Now, if you would like to undo this, if we are breaking this molecule down, we can break this peptide bond, boop, boop, by adding water back, and you will notice that our H and our OH are back where they started. Now, we tend to spend a little bit more time on proteins because you have a lot of proteins in your body. Okay, so if we go back to our list, we've got enzymes, structural ones, movement, defenses, fuel. We've got so many different functions. Okay, we tend to spend a little bit more time on proteins because of all the functions, but it's also really important to talk about what happens if you break some of these proteins. Okay, so you can do what's called denaturation. And basically, your fully functional proteins have a very specific shape. Okay, and each shape is unique to that particular type of protein. And that protein only functions with the correct shape. If you change the shape, you change the function. If you change the shape enough, you lose function altogether. Okay, and there are different ways to change the shape. You can get your proteins too hot. You can change the pH too much, so too acidic or too basic of an environment. There are certain types of chemicals that are denaturing chemicals. They can break your proteins. Okay, you could even do things to disrupt the hydrogen bonds that help stabilize some of the protein shapes. So if we're breaking proteins, what are we having ultimate effects on? Well, what were we doing to start with? Enzymes, structural roles, movements, defenses, okay? All of the enzymes in your body. Remember we said we could do chemical reactions without enzymes? We just don't like to. It goes too slow, we use too much energy. Well, if you break all of your proteins, if you denature them all, your enzymes don't work. And then guess what happens? You don't work. Okay, so heat, one of the things that's easy to think about because everybody gets hot. What is the difference between you getting a fever of 100 degrees versus you getting a fever of 105 degrees? Okay, everybody knows if you get a fever of 100 degrees, okay, then you're probably taking some Tylenol, you're probably spending the afternoon in bed just trying to recuperate and giving your body a chance to mend itself. If you get a fever of 105 degrees, you're going to the emergency room. What's the difference? It's just 5 degrees. Okay? If you break your proteins, you're not going to live. Okay? So if your temperature gets too high and your body cannot get back to homeostasis like it's supposed to, you need medical intervention sooner rather than later. We do not want to denature our enzymes. Now, because we have so many different functions for proteins, it will probably not come as a shock to you that we also have different shapes. We did mention that the shape dictates the function, so we have two options here. We have fibrous proteins that look more like ropes or strings, and then we have globular proteins that kind of look like blobs. We're not judging, okay? Fibrous proteins, these long skinny rope-like ones, tend to be composed of mostly nonpolar amino acids, and just like a rope would, we're adding strength and durability to certain structures. So you'll find a lot of these in like your cytoskeleton, your tissues, especially your skin tissues that are giving it some shape. And then your globular tissues, these blobs. Okay, these tend to be composed of more polar amino acids, with the polars being all over the outsides, exposed to the watery environment of your body fluids. These are the ones that tend to function as enzymes, hormones, and other cell messengers. These are just easier for your body to transport throughout your body fluids. And so your hormones, if you know about your hormones, they travel through your blood. Okay, that's a watery environment. We're going to want some sort of polar molecule or a hydrophilic molecule, so there's no negative interaction there. Okay, so how do we get to that shape? We have to fiddle with our little amino acids here. Okay, so we talked about taking amino acids and peptide bonding them together. We're going to make our polypeptide. That is what is referred to as your primary structure. It's just the amino acid polypeptide chain all by itself. Okay? Not a whole lot of shape there, so we're going to have to start modifying this. Your secondary structure comes from you folding or twisting your amino acid chain. So you can create what are referred to as beta pleated sheets or alpha helices. Now, most people have heard the term helix. We usually refer to your DNA as a double helix, so there would be two strands. So that shape is somewhat familiar to us. A beta pleated sheet, That's a little hard to describe. If you think about taking a piece of paper and folding it over and over and over on itself to create a fan, like a little child would do. If you kind of imagine that shape of that fan, the folded paper, that's what a beta pleated sheet kind of looks like. Now, we're starting to add some shape, but if we just stop here, this can easily kind of unfold itself and go right back to our chain. So we need to do at least one more step, tertiary structure. This is that overall three-dimensional shape that we are creating. We're further twisting and folding and coiling. Okay? We are stabilizing with hydrogen bonds. We might even create some disulfide bonds to help really solidify this overall shape. Okay? And a lot of proteins will stop there. Not everyone though, but a lot of proteins will just stop there. And this is perfectly fine. If you are a protein that only needs a tertiary structure, once you have coiled and bonded, you're ready to go. You're ready to be functional. However, some proteins require a quaternary structure. This comes into play when you have multiple subunits or multiple pieces that have to be joined together before your protein is fully functional. So notice our globular protein, we stuck a couple of blobs together. Okay. Our fibrous protein, notice that we stuck a couple of ropes together. If you are a protein that requires a quaternary structure to be fully functional, you will not be fully functional until you have all of your components. Okay, our last organic molecule, okay, nucleic acids. The monomers of nucleic acids are nucleotides. Okay. You have three parts to a nucleotide. You have a nitrogenous base. You have a 5-carbon sugar. And you have a phosphate group. Okay. If you know anything about your DNA and your RNA, it's a deoxyribonucleic acid. Ribonucleic acid. Okay. Your DNA and your RNA are composed of repeating nucleotides. Okay. Now your bases, we have 5 options. If we are talking about DNA, we have adenine, guanine, cytosine, and thymine. If we are talking about RNA, we have adenine, guanine, cytosine, and uracil instead. Okay. They all have fairly similar structures. Little slight differences here and there. Okay. Next, we would be bound to a 5-carbon sugar. Okay. Could be pentose, could be ribose, could be deoxyribose. Right? 5 carbons, just slightly different add-ons. And our third component is a phosphate group. Now, a phosphate group is a phosphate group. They all look the same. It is not until you put all 3 of these pieces together that you have formed one nucleotide. And then we take each of these nucleotides and we attach and we attach and we attach. Okay? So each of these little sections, okay, you can see the colors. Each of these little sections is its own nucleotide. And it's just repeating unit after repeating unit. That's when you form your nucleic acids. Okay, so your DNA is a double strand of nucleotides. Your RNA is a single strand of nucleotides. And we do have one more molecule up here, ATP. Okay, so adenosine triphosphate, adenosine triphosphate, adenosine adenine, okay, is one of those bases that we just talked about. We are going to attach that to a ribose sugar, that 5-carbon sugar. And then not only do we have 1 phosphate or 2 phosphates, but we have 3 phosphate groups. That's where we get the triphosphate from. Okay. This is our absolute 100% favorite source of energy in our bodies. If we are talking energy, there is a 99.999% chance we are talking about ATP. Okay. Now, once we use ATP, the first thing we do is chop off a phosphate group. You are left with adenosine diphosphate. This does still have a little bit of energy left. You could chop off that second phosphate group, be left with adenosine monophosphate, AMP. Okay. And you might think, oh, AMP can't be super useful. You would be wrong, for better or for worse. Okay. We can actually take AMP, modify it just a tiny little bit, turn it into something called CAMP. Okay. That is one of the ways that your hormones perform their duties. They use CAMP to activate transcription and translation, or protein synthesis, inside your cells. So AMP has its uses too. Okay. So don't just think it's all about ATP, maybe a little ADP here and there. All of these are useful in their own right. And that concludes our lecture on organic molecules.